We live in a world our ancestors would barely recognize. Around the globe, the impact of human ingenuity is now everywhere. We push back the limits of our planet at speeds, depths, and heights that would have left our forebears breathless. Driving all these achievements is humankind's extraordinary gift for invention. Through genius and inspiration, we created exceptional solutions to complex problems, from the everyday to the spectacular, some good and some not so good. This series celebrates the million ways our great inventions have transformed our world.
It's one small step for man, one giant leap for mankind.
Perhaps no single invention has changed mankind more fundamentally. It's a technology that reimagines our future. It helps feed a growing population and heal diseases. It even holds out the promise of eternal life.
I can't think of a single sector in which it will not play a significant role in the future.
It already influences our daily life without us even suspecting it.
The future's really promising. To these scientists, hardly anything seems impossible.
But many people are understandably afraid of losing control. How do we decide what should be allowed and shouldn't be allowed?
Opposition is mounting. It knows no limits. It makes possible things that still sound like science fiction. Its applications are rapidly expanding, and it's unstoppable.
Many things we would have found hard to imagine are now absolutely possible.
Humankind has learned to read the code of life, and now it's learning to rewrite it.
I think in many ways the genie is out of the bottle. There's no way back.
Thanks to genetic engineering, we're at the cusp of a new evolution. Two billion years ago, the planet's first living beings appeared in the form of single-celled organisms. During the course of evolution, they flourished and spread all over the globe. All living beings are identical when it comes to the way the blueprint of life is stored and inherited through cells from one generation to the next.
We have billions of people living on this planet, and even though our genome differs from everyone else's by only 0.1% or so, the fascinating thing is that no human is identical to any other human in the world. Each person is a true individual.
Genes are the key to life, but also the key to illnesses. Genes decide whether a single cell develops into a tomato or into a human being, and they decide whether I'm good at maths and whether my hair is straight or curly.
Genetic engineering makes it possible to decode and rewrite the genetic makeup of living organisms. Living beings can now be altered and recreated. Without doubt, genetic engineering is one of mankind's most momentous discoveries ever. It sounds lab-like and futuristic, but it has already become routine. Our food labels rarely state the presence of genetically modified ingredients. In actual fact, however, only a few products in our supermarkets haven't been touched by genetic engineering.
80% of our soy crops are genetically modified. They're used in part of animal feed, and so that gets into our meat and dairy products. For instance, pork gelatin is used in confectionery and fruit juice. We eat food every day: bread, meat, cheese, salad dressing, soft drinks. About 70% of all our daily groceries are genetically modified in some way, and most people don't have a clue.
As the global population surges, some see genetically modified foods as the only way of preventing food shortages in the future. By 2050, probably more than 9 billion people will need to be fed. That's 2 billion more than today. But the food industry is not the only area where genetic engineering impacts our lives. Genetically modified cotton has conquered global markets. The fashions that are presented on the catwalks of New York, Paris or Milan can be found within just days as cheap copies on our high streets. Whereas there used to be only 2 collections annually, major textile companies now produce up to 24 collections a year.
Shopping is part of our modern lifestyles. So inner cities are bursting with shops. Between 2002 and 2015, clothes sales actually doubled. And the future says that this tendency will only continue to rise.
Cotton is a crucial material. I mean, it's so fantastic. You can wear it in summer or winter. You can layer it because it's breathable and comfortable. So it's no surprise that it's been successful. And since the 1980s, cotton sales have doubled, and genetic engineering has played a major role in that process.
People were already cultivating cotton 7,000 years ago. Cotton was one of the engines of industrialization and globalization. Demand for it is so great that crop yields need to be steadily increased. And to effect this increase, cotton plants are genetically engineered.
76% of cotton is already genetically modified. There's hardly a wardrobe in the world that doesn't contain cloth from a genetic engineered cotton.
We pay for this clothing with credit cards or cash. But in most countries today, even banknotes are still made of cotton. And it's practically always genetically modified. Cotton is temperature insensitive and tear-proof. It's very suitable for long-time use. Our clothing is washed at home with laundry detergent, some of whose latest improvements go back to genetic laboratories. In fact, practically all our planet's detergents were developed with the help of genetic engineering. But it wasn't always like that. Until the 1950s, doing the laundry was largely seen as women's work. This was hard, physically demanding, and time-consuming. Wearing a fresh set of clothes every day was unthinkable under these circumstances.
The kind of clean you like best, next to those you love.
Up until the early 1980s, detergents still contained lots of environmentally harmful phosphate. An enzyme synthesized through genetic engineering was finally able to tackle this problem. Enzymes remove stains from laundry very efficiently.
The cleanest clean under the sun.
When I tell my mum that her laundry detergent contains enzymes that are a product of genetic engineering, she thinks I'm mad. To her, genetic engineering is an abstract technology that has something to do with soybeans and corn.
Without us noticing, it's become part of all realms of our daily life.
The field of genetic engineering continues to evolve. In the meantime, it stops at nothing and keeps expanding the frontiers. Legislation on what is and isn't acceptable has long been outpaced by the march of science. Aqua Bounty salmon is genetically modified. Normally, salmon doesn't produce growth hormones in winter, so scientists added a growth gene and an anti-freeze gene from other fish species. So now the salmon continues to grow even during the cold season. Not only does it grow faster, it also grows substantially larger than natural fellow species. A lucrative outlook for breeders. Steadily growing quantities of inexpensive animal products, such as fish or meat, are leading to the over-fertilization of our oceans, rising greenhouse gas emissions, and the excessive use of antibiotics. But how else can we get a handle on the ever-rising global demand for meat and fish? An international group of experts was invited to witness a special event in the television studio. A burger was being presented. With a price tag of $300,000, probably the world's most expensive burger, a prototype. Mark Post from the University of Maastricht had extracted stem cells from bovine muscles that he was able to grow into thin strips of muscle fibre. Some 20,000 of these cultured muscle fibres were compressed to make this burger.
The first time ever that we're going to be making meat from cow cells, but this meat is not coming from inside a cow, it's growing outside of it. And I mean, you can make a hamburger with it, you can fry it and eat it, and it will taste just like normal beef, but without all the environmental and animal suffering downsides.
You need nutrients to make lab-grown meat, and these are obtained through genetically modified microorganisms. This means factory farming may one day be a thing of the past. Right now, however, the meat without animals business model is still too complex and expensive.
I think that eating meat out of a laboratory would actually be quite a positive thing. You know, the notion that you're eating something kind of created in a sterile, clean way that's the healthiest meat possible, rather than eating something that you know an animal suffered from, that an animal probably suffered its whole life from, that was sort of raised in conditions that you don't know about. I think people kind of initially sort of balk at the idea, but actually, I think if we get it right, artificial meat would be definitely a better thing, both for the individual and for the environment.
It would look like meat, taste like meat, but wouldn't be meat. A global competition is on to be the first to market it. Many large international corporations from the meat and food processing industry are working feverishly on this new product. It promises to be $1 billion business.
When it comes to meat from a lab, I see a definite advantage for our environment. Animal-based foods are not very eco-friendly because they require about 3 to 4 times as much land, water, pest control and labour than plant-based foods. From an environmental point of view, the project is certainly desirable.
Genetically modified organisms, or GMOs for short, are increasingly shaping our diet. But the public is still wary. A large majority of people reject GMO foods. There are too many concerns about potential effects on human health and the environment. This also applies to plant breeding. Selective breeding can be a long and arduous route. For centuries, different varieties have been crossbred to either add positive characteristics or to remove unwanted ones. In doing so, people were constantly confronted with the problem of how to accelerate breeding and improve yields. In the 1950s, atomic energy gave plant breeders a completely new idea. Associated with a number of horrible events of the 20th century, the atomic bomb, the Cold War's nuclear threat, and nuclear reactor disasters of Chernobyl, and now also Fukushima, nuclear power is still on the defensive today. But it wasn't always like that. Nuclear radiation once stood for a bright and exciting future. In the 1950s, atomic radiation technology was thought to hold great promise for just about everything. It was considered a magic weapon in seed breeding. Irradiated rice or irradiated tomatoes were a big hit with consumers. Atomic or gamma gardens were en vogue. Highly aggressive radiation was meant to cause random mutations that would give rise to new plant varieties. In the early days of atomic gardening, irradiation was carried out directly in the field.
What sounds unthinkable for us today is actually still common practice. Of course, this is done in high-security labs, but hardly anyone knows that genetically modified rice, wheat, oat, rape, maize and soy are produced using radiation from radioactive sources. Peanuts too, as well as beans, many types of fruit and vegetables, they use this method.
In Vienna, seeds are bred in a lab run by the International Atomic Energy Agency. For 50 years now, scientists have been irradiating seeds to make them more resistant and give higher yields. Across the world, almost 3,300 new varieties have been bred in this way.
Many of the modern wheat varieties that we have today are a result of genetic modification thanks to radiation. And in fact, A large amount of the durum wheat that we use to create pasta was originally bred in this way. I think it's kind of cool.
Atomic gardening gave rise to thousands of new varieties. People all over the world love beer. Yet few know that practically the entire barley harvest of Europe, which is needed for brewing beer, carries one of two modified genes. Intentionally exposed to radiation decades ago, the resultant mutation led to shorter, more robust stems to carry the ears.
The vegetables and fruit we know nowadays, for the most part, have nothing to do with their natural origins. The wild potato was toxic, which was its way of protecting itself from insects. The original cucumber, that was small and bitter. The original banana was green, small, and contained hard seeds.
The breeder's aim was always the same: enhanced nutrition and greater yields. Genetic engineering made this easy. In 1983, molecular biologist Jeff Shell achieves a decisive scientific breakthrough. At the Max Planck Institute in Cologne, he's the first to insert foreign genes into a plant cell. A characteristic of one particular organism is transferred to a host plant. The starting pistol had been fired. What previously took hundreds of years can suddenly be done in no time at all.
This opened up enormous possibilities, of course. Being able to transfer traits from one organism to another meant that one could now actually start creating completely new kinds of plants.
In the spring of 1996, a few US farmers sowed genetically modified seeds for the very first time. Quite a historic moment in retrospect, especially if you consider how much the technology has spread ever since.
Genetic engineering is cracking the code of life. But it's been a long and bumpy road. How life's building blocks are passed on from one generation to the next has puzzled us for thousands of years. A monk succeeded in unraveling the great mystery. At first, no one took note of his discovery, but then it hit like a bombshell. For thousands of years, people have been mystified about the workings of heredity. The Greek scholar Pythagoras believed that semen coursed through a man's body while picking up hereditary information that's passed on to the woman. Aristotle assumed that the building instruction for a new human came exclusively from men, and also believed that a woman's building material consisted of her menstrual blood. The Swiss Theophrastus Bombast von Hohenheim, aka Paracelsus, was one of the most famous physicians of 16th century Europe.
To Paracelsus, previous theories seemed too complicated. He didn't need women for his genetic theory. He believed that there was already a miniature human in the man's sperm, huddled, waiting to grow up into a baby.
Women played the role of incubators. Paracelsus was convinced that sperm alone produced human beings. In fact, he believed that semen could be warmed with horse manure, buried for 40 weeks, and still result in a human being, though with one small impairment. This horse dung-grown human, Paracelsus believed, would probably exhibit monstrous characteristics. The foundation stone for today's genetic engineering was laid in the then Austrian city of Brunn in 1865. It's where Gregor Johann Mendel lived. He's the Augustinian monk who used peas to derive the laws of inheritance. Mendel's laws are the first scientifically based theory of inheritance and thus the foundation of genetic engineering. He analyzed 28,000 plants to prove his data. Mendel observed that certain traits were dominant and these asserted themselves, while other traits were recessive and were not expressed.
Ironically, Mendel wanted to become a teacher, but he failed the exams twice. And in biology, of all subjects, the very field which he would later make such a substantial contribution.
The tragic side to this is that people didn't really regard him as a scientist during his lifetime. To his peers, he was more like a hobby breeder, a monk with odd interests.
And so the results of his research didn't interest anyone.
It's tragic because this work was his lifetime achievement. Following its publication, his article on his findings was only quoted 4 times. Hardly anyone took any notice of it. He was simply forgotten.
Mendel died in 1884 of kidney failure, and his writings were only discovered by chance, so he narrowly missed out on making history. After his death, a number of scientists continued to obsessively examine in parallel the mystery of heredity. They had no clue that the results were already in print at a library in Brünn, Mendel's Laws.
The scientific world was forced to admit that not only was it 35 years late, it had also unjustly disregarded and ignored the work of a brilliant monk from Brunei.
Mendel's laws were a ticking time bomb. What would happen if they were to be applied to humans? In the late 19th century, the Cecil Hotel in London boasted 800 rooms. It was Europe's most imposing hotel, and also where, in 1912, the first International Eugenics Conference was hosted. The assembled eugenicists wanted to eradicate undesirable hereditary traits that are passed down through reproduction. In line with their twisted beliefs, this, they thought, would create a healthier society. Forced sterilizations and the internment of genetically unsuitable individuals were used to weed out the weak, the sick and the insane. The conference was a dark foreboding of times to come. In Germany, the Nazis turned theory into reality. They exceeded the eugenic ideals with ruthless brutality and cruelty. To them, anything was justified to create the new Germanic master race. In 1940 and 1941, 70,000 people with physical, mental and emotional disabilities were systematically murdered in gas chambers. And the killing continued in their psychiatric institutions and hospitals. It took the collapse of the Third Reich in 1945 to put an end to the horror of the Nazis. It still wasn't possible to read the genetic makeup of living beings, and science was far from being able to modify the genome in a targeted way. The last piece of the puzzle that scientists were still missing was the structure of our genetic material, DNA. The 1950s. The war is over, and people are longing for peace, security, and modest prosperity. Freezer technology, convenient and now also affordable, is embraced. The VW Beetle is one of the best-selling cars, and televisions are entering living rooms. And when it comes to genetics, the science community is on the verge of the biggest breakthrough ever. James Watson and Francis Crick meet each other at a lab in Cambridge. They're intent on unraveling the mystery of DNA structure, whatever it takes.
James Watson and Francis Crick knew that if they could identify the structure of DNA, this would be a milestone in the history of science. It was the million-dollar question.
Many were working on ways to find the answer. Competition was high. Watson and Crick were obsessed with the topic, and they were both very ambitious. They both knew that only the very first to present the solution would get all the honour and the glory.
But there's one particular rival that wasn't on Watson and Crick's radar. London's King's College isn't far from Cambridge. This is where Rosalind Franklin, a scientist, is working to unlock the exact same mystery, the structure of DNA. Rosalind Franklin was a biochemist, but as a woman and a scientist in the 50s, she struggled in a male-dominated workplace. In the 50s, women tended to their husbands, children, gardens, and their appearance. In the science world of those years, they mainly filled the role of assistants or secretaries, making coffee and supporting their male colleagues in the laboratory.
Despite being an excellent scientist, Rosalind unfortunately suffered from the sexist ways of her time back then. I mean, it was, from all accounts, very normal for male scientists to make insinuating remarks about a woman in her presence. And in fact, it was also normal that the male scientists would meet in clubs after work to discuss their findings, but women weren't granted access to those same clubs.
At King's College, Rosalind Franklin was studying X-ray diffraction when she managed to take one of the most important photos in the history of science, Photo 51. It shows the structure of genetic material, or DNA, and then something unexpected occurred. James Watson happened to be at King's College while making a visit to London.
One of Franklin's colleagues secretly showed the crucial photo to James Watson, an absolute no-go in the science world. Watson immediately understood that the structure of DNA was a double helix, and that Rosalind Franklin was ahead of them in the race.
In April 1953, Rosalind Franklin presented her findings on DNA, and at the same time, so did James Watson and Francis Crick. The results were identical. Sadly, she died too early.
In 1962, Watson and Crick were awarded a Nobel Prize for their discovery of DNA structure. Rosalind Franklin had died 4 years previously. During their acceptance speeches, Crick and Watson failed to mention Rosalind Franklin. She was given no credit whatsoever.
The discovery of the double helix structure of DNA was a true turning point in the history of science, comparable to the Copernican revolution. It changed everything.
DNA has a twisted ladder shape, and each rung consists of interlinked chemical building blocks. This genetic code can be likened to a computer code that directs all the cell's actions.
Picture this. If you could take the entire length of the DNA strands within your body and make them into a long single strand, it would stretch as far as the Earth to the Sun, 100 times over. And every human being contains this DNA inside them. It's utterly incredible what nature is capable of producing.
Our knowledge of DNA structure is a stepping stone that enables us to read and rewrite the code of life. It even changed the work of criminal investigators. Prior to this revelation, murderers could simply vanish into thin air. And yet another discovery made it possible to even go back and reopen old, unsolved murder cases. Day for day, we shed about 10 grams of dead skin cell flakes. Traces can be found on our toothbrush, comb, and clothes. Each individual has their own unique DNA that stores all their genetic information. So we leave a DNA footprint wherever we go. The person who discovered DNA fingerprinting is Alec Jeffreys. He stumbled upon this technique by chance in 1984, and it revolutionized forensic science.
The discovery of the DNA trace, or genetic fingerprinting, is a fundamental milestone in forensics. Even the smallest piece of dandruff can convict a perpetrator. It's become almost impossible to not leave a trace.
But other spectacular methods have also become available to scientists. In the fall of 1998, a pregnant woman is brutally murdered on the verge of a highway in the Netherlands and dragged into the forest. A walker finds the body under a cover of oak leaves. The victim's husband falls under suspicion, but denies ever having been at the crime scene. The case is reopened 6 years later. Shortly after the murder, investigators have found an oak leaf in the husband's car. Perhaps it can provide new information. It's sent to the German Federal Criminal Police Office for the world's first comprehensive DNA analysis of a plant connected to a murder case. This had never been done before. The leaf's DNA gives scientists an important clue. It's from an English oak. But where can this particular tree be found? An investigation is started to compare the secured DNA with that of all the relevant oaks at the crime scene. Even years later, they're able to assign this leaf to a specific oak tree. For the first time ever, the court admits the leaf, along with the new forensic method, as evidence. The husband's lies are exposed, and he is convicted of murder. Genetic engineering rapidly gains momentum, and many new ideas pop up. Genetic engineering is a buzz with a gold rush atmosphere. It becomes a big business. Anything that's possible is also tried. With the help of genetic engineering, scientists can not only stop the dramatic extinction of animal species through cloning, they could also bring extinct animals back to life. Will dinosaurs be de-extincted in the future? Will we bring people back to life one day? July 5th, 1996 is an important day in the history of genetic engineering. It's the day Dolly the sheep, the first mammal to be successfully cloned, is born.
I remember when those images of Dolly, the world's first cloned animal, went around the world. And I mean, she was such a cute, innocent little sheep. But she's no longer unique. By now, over 20 different animal species have been cloned. We've seen pigs, dogs, cats, cows, even monkeys. Cloning has become a normal practice in labs around the world.
It's well known that the love for a pet can be boundless. Gunther, a sheepdog, inherited $140 million from his owner, Countess Carlotta von Liebenstein. He now owns several valuable estates and keeps several domestic servants busy with his care. True love knows no financial limits. So just imagine how much money people would be willing to pay to make their pet immortal. A South Korean biotech company has a lucrative business idea. They're willing to bring back people's beloved pet dogs as cloned copies for $100,000. The replicated puppies are raised in sterile kennels where they receive round-the-clock medical care.
Cloning is an exclusive business that currently is only taking place in social classes for which money is not an issue. No normal person would consider cloning their pet, but the cost will probably decrease over time, and one day this might be a normal thing to do. I can imagine that clone labs will eventually start cooperating with vets.
As always, the market is going to regulate itself. If there's demand for pink mice that glow in the dark, there'll definitely be a business that will start producing them genetically, providing it's legal. That's just the way our system works.
To clone a much-loved pet dog, all the laboratory needs is a piece of skin or connective tissue. Commercial providers have ready-to-use kits that make the extraction process very simple. The punched-out tissue sample can be already archived in nitrogen during the dog's lifetime. This way, the clone can be created right after the pet dies.
There's no limit to genetic engineering. There's a profundity of what we can do, not just in terms of solving for illnesses, but actually the actual species that live on the world, right? So up until now, it's been about waiting for evolution to happen, waiting for those changes to happen naturally. What genetic engineering does is it means it puts humans in the driver's seat and allows for those changes not only to happen much more quickly, but to take a different direction than maybe they would have naturally.
Even if we as a society say no, we don't want that to happen, there still might be someone somewhere in the world who'll go ahead and do it anyway. There's no way of controlling it.
Genetic engineering has become a big business. The world's largest animal cloning factory is currently being built in the Chinese megacity of Tianjin, which has a population of 14 million. High-quality bovine embryos will be produced here to satisfy the rapidly growing demand in China for high-quality beef. This business has made inroads into professional sports as well. In Argentina, cloned horses are all the rage. Polo is one of the most popular sports here, and since world champion ponies are neutered, they cannot pass on their winning genes. Now, particularly successful polo horses are cloned at embryo farms. Former champions are produced serially. So, wouldn't it be theoretically possible to resurrect, as a clone, a person who died in a car accident? So far, humans have never been cloned, but many scientists believe it's been technically possible for some time. In January 2018, Chinese scientists achieved another breakthrough. They cloned 2 monkeys for the first time ever.
In the so-called pyramid of life, we do classify monkeys as part of the upper ranks. You know, they belong to our closer relatives, so they're different to cats and dogs, which means that clone research is approaching humans step by step. We will be facing big questions really soon. Where is this all going to lead us? That's the biggest question.
Boston. Molecular biologists at Harvard University are hoping to bring Neanderthals back to life. In 2010, scientists managed to decipher the Neanderthal's entire genome. Neanderthals became extinct thousands of years ago. The Neanderthal's DNA has fragmented into many small pieces. There are no intact DNA molecules left from which he could be resurrected. The idea scientists have is to take a fertilized human egg cell and rewrite it with the remnant DNA of a Neanderthal. Technically, it's possible, but a surrogate mother would be needed to carry our extinct relative.
Up until now, everything that has eventually become possible has simply been done. Can we do it? Then let's do it. But if I picture Neanderthals suddenly roaming around the streets, that's a little more unsettling, like a sci-fi movie come alive. And I wonder what we do with them. Put them in a zoo? Put them up in a council flat? Is that ethical?
Cloning dinosaurs for a theme park would be a sensation. But this is where the almost unlimited potential of genetic engineering ends. The problem is, the DNA decomposes after 1 million years, but the last dinosaurs lived 65 million years ago. So this idea will always remain pure fantasy.
The boundaries are blurring between science and scientific playground. If scientists are now trying to bring extinct species back to life, is that just a demonstration, like, hey, look what we can do? Or is it actually a way of bringing back extinct species to promote biodiversity? I suppose it's both.
There are no distinct boundaries between play and science. Scientists have discovered something of use for society, but should this preclude it from being commercially exploited? What matters most is what we need, and millions of people are waiting for a donor organ, for example. Genetic engineering could help solve this problem. Decoding the genome brought to light surprising findings. Genetically, we humans are 50% identical with bananas and 99% identical with monkeys. And even if pigs differ from us in appearance, and particularly in behavior, in one important aspect, we are similar: our organs.
I think it's fascinating how we as creatures are all so similar to one another.
The idea of implanting animal organs in humans is not new. Monkeys in particular were popular candidates for this. Serge Voronov, a doctor, was famous for this. In the 1920s, he performed rejuvenation surgery on older men by implanting sliced chimpanzee testicles into them. The demand was huge. Older men were hoping to recover their youth and potency. The grafting of chimp testes to humans set a precedent. Global demand for this procedure was so great that occasional chimpanzee shortages were experienced. Thousands of operations were performed, and Voronov was highly esteemed by colleagues until it emerged that the positive results were just a placebo effect.
It's quite intriguing that many of today's ideas are not entirely new. Take the idea of transplanting animal organs into humans. Several hundred years ago, people were trying to do just that. In the 17th century, people were receiving blood transfusions from lambs or skin transplants from frogs. It wasn't that the ideas weren't around, there just wasn't enough knowledge to do these things successfully at the time.
Nowadays, humankind has found a way to do it. We're mixing species back and forth. Genetic engineering has made this a normal practice.
Millions across the world are waiting for a donor organ. While pursuing a solution, scientists had a crazy idea. And now, with the help of genetic engineering, it may well become a reality one day. At Harvard's medical school, they're working on pigs that one day will serve as organ donors for humans. The special thing about pigs is that their organs are similar in size and shape to human organs.
The need for kidneys and lungs and hearts and intestines, it's dramatic. Some people wait for literal years and die as a result. So if we can cultivate these and grow them, it could save thousands, not millions of lives.
The challenge is to genetically modify pigs in such a way that the human body no longer recognizes their tissue as foreign. To this end, embryonic pig cells are manipulated. And hopefully these cells give rise to offspring with organs that won't be rejected by the human immune system.
About 200 years ago, whalers and sealers released pigs on the Auckland Islands in New Zealand. These pigs are particularly suitable as organ donors because their breed lived in isolation on the islands for a long time without any contact to humans. That's why they have less viruses.
The precondition for successful organ transplantation is virus-free pigs. Porcine viruses pose an unpredictable risk for humans. Editing out these viruses is expensive and time-consuming. But from where else can urgently needed donor organs be obtained? In 2015 in San Francisco, actress Cameron Diaz is given the honor of awarding one of the world's most valuable science prizes, the Breakthrough Prize. Worth $3 million, it's donated by Facebook founder Mark Zuckerberg, among others. Emmanuelle Charpentier and Jennifer Doudna are honored for their groundbreaking CRISPR-Cas9 gene editing tool, also known as gene scissors. CRISPR gene scissors triggered a scientific revolution and catapulted the world into a new frontier of genetic engineering. CRISPR lets us reprogram nature. These days, you can read about the successful application of gene scissors almost every day in the news.
It's really a transformative technology in the sense that it allows now the manipulation of the genomes in a precise manner, but in a very easy and versatile fashion, and also at low cost.
The technology has been embraced by the science world because it works in all cells and organisms.
CRISPR is the most groundbreaking discovery of the past decade. By now, CRISPR is the most used genetic engineering tool in labs across the whole world. We can't tell where this technology will lead us in the future, but we know it's very powerful.
The incredible thing about CRISPR is that it's not only relatively simple to use, but it's also become very low cost. You don't need a high-tech lab in order to use it. And this opens up a realm of possibilities. Obviously, a lot of great benefits can happen from this democratization of technology, but it also carries a lot of risks too. And so it's an interesting new world we're entering into.
CRISPR works like a set of gene scissors with a target indication. It finds the exact target site of the DNA, and the protein Cas9 docks onto it and cuts.
CRISPR is a little like a genetic pair of scissors. You can rewrite genes as you would rewrite text in a word program. They can delete a gene or insert another. It's so easy that after a short crash course on it, even pupils can do it.
These fish are not found in the natural world. You can only buy them online. Thanks to genetic modification using CRISPR, these fluorescent fish can be ordered in sunburst orange, electric green or starfire red for home aquariums. Gene-edited creations have taken on a life of their own in labs around the world, including mice whose DNA is inserted with a green fluorescent protein derived from jellyfish.
With CRISPR, nature has become like a malleable game that can be changed to one's liking. The question is who determines the rules and where are the breaks?
The first genetically modified humans were brought into the world in China. They are twins who are immune to HIV thanks to CRISPR. Their creator, the scientist, He Jiankui, was sentenced to 3 years in prison for illegal genetic manipulation of human embryos.
We've opened Pandora's box. I mean, the genie is out of the bottle and we can't get it back in very easily. And I mean, I think it's part of human nature. Everyone understandably wants to be better, wants to be healthier, and they want to be a winner. So it's understandable that it'll be adopted.
It might be up to the labs one day to decide on our talents, our looks, our intelligence. And we're just at the beginning of this whole new era.
CRISPR gene scissors are inspiring scientists and turbocharging genetic engineering. Things that took decades using conventional genetic engineering can now be done in short order.
The discovery of CRISPR is a revolution because it allows science to work at least 1,000 times faster, more efficiently, and more cost-effectively.
At Harvard's medical school in Boston, scientists have achieved a breakthrough thanks to CRISPR technology. Gene editing was able to eliminate the porcine viruses that endanger humans. This has brought the pig a big step closer to being an organ donor. One day, it may be possible for humans to live with pig hearts. CRISPR gene editing has made it quick and easy to modify or even create new living beings as we please in labs. But what would happen if genetically modified organisms suddenly start to change and propagate themselves? No creature is responsible for as many deaths as the mosquito. Malaria alone kills 500,000 people every year. Dengue fever, the Zika virus, and chikungunya fever are also spread by mosquitoes. These diseases could only be eradicated by killing off all the disease-transmitting mosquitoes where such diseases are endemic. But even the widespread use of pesticides, which are harmful to the environment and pose a health risk, cannot prevent the spread of mosquitoes. Scientists at MIT in Boston came up with an idea. CRISPR gene editing gave them the tool they need to genetically modify mosquitoes, thereby overriding the rules of heredity. The goal is to defeat malaria by forcing disease-transmitting mosquito populations into extinction. They use a technique called gene drive, whereby some mosquitoes are genetically manipulated to render their offspring infertile. To make this work, a turbo is added to the modified gene. Even just a few gene mosquitoes released into the wild would suffice to wipe out entire populations within 1 or 2 years. But what happens if the plan goes differently than expected?
I hate mosquitoes for all kinds of obvious reasons, but on top of that, malaria is one of the biggest killers in the world, and so it's incredibly important that we explore things like gene drives to reduce the risk of malaria. But at the same time, we do need to be careful that there aren't some important role that they play in keeping a healthy ecosystem.
MIT actually filed for a patent on the gene drive to prevent its misuse because it's so dangerous. The gene drive can't easily be reversed, and so far, gene drive experiments have not been conducted in the wild. There's considerable concern that such experiments could get out of control. Nevertheless, scientists are hoping to use this method to eradicate malaria in the future.
If you ask me, it's a little bit irresponsible to do something like that without knowing how to reverse it. But people have different opinions on it, and it just makes me wonder who's going to be setting the rules in the future.
Since the advent of CRISPR gene editing, developments are progressing faster than scientists thought possible. In the future, biohackers could not only alter traditional pathogens and weaponize them in wars, they could also modify insects, including locusts, to transmit diseases and destroy crops. Entire nations could be brought to a standstill and harvests destroyed. This may sound like science fiction, but defense ministries are seriously exploring these kinds of emergency scenarios. Genetic engineering is becoming more and more prevalent in our lives. Its influence is growing, and hopes and expectations tied to this technology are mounting. It's expected to boost crop yields and help these defy climate change, feed a growing world population, cure diseases, and combat malaria. Already now, genetic engineering is solving many problems. In the future, it will solve problems that we can't fathom today.
Genetic engineering may well make our lives better, but it's really up to us how we manage this remarkable tool.
Genetic engineering has come a long way from the time the father of genetic engineering, Gregor Mendel, conducted his pea experiments. Many pioneering discoveries have paved the way for unimaginable things. The pace of research in genetic engineering has increased 1,000-fold.
We have to ask ourselves as a society, Where do we want this to lead us?
Genetic engineering is in its infancy. Much of its future lies ahead of it. Nobody knows how the world of tomorrow will look. Only one thing is certain. There's never going to be a world without genetic engineering.